Hi everyone, so this is a virtual lab for AP Biology Investigation 6, which covers the topic of cellular respiration, and the way that we do that is using a respirometer. So a respirometer, and to be honest with you, this is probably a lab that has been asking to be made virtual for a very long time. I find it to be incredibly complicated to run in a high school lab environment. But some teachers do still physically run it. For me, I essentially have always run it virtually or through a demonstration. But the respirometer has several iterations. So the old school respirometers are a large glass vial with a stopper at the top, and inside the stopper a hole is drilled so that you can fit a 1 ml pipette. They've made some newer ones that are smaller micro respirometers. I know some people that use them, but many people still don't use them. But we're going to run through this in a virtual fashion. So just imagine, and I will have showed you in class for my students what a respirometer is. But the important part of it, it's all important. But inside this glass vial, you can fit components of the experiment, and then this 1 ml pipette is going to act as a gauge for gas consumption. So decreasing volumes of gas can be read on this 1 ml pipette. So, basically, if you're going from 0.9 to 0.8 to 0.7, that's a 0.2 milliliter difference. So we'll go over that in a minute. But you can actually gauge the amount of oxygen gas consumed using this very simple setup. And basically what it is, excuse me, for the respirometer, we're going to take trays of water and submerge the respirometer into the water. And what happens is water rushes into the tip of the respirometer up to a certain point, and from there on is the air from the glass vial pushing back against that water bubble. And so we're going to set up this experiment such that whatever organism we're studying the respiration rates of will be in the glass vial, and as that organism performs respiration and consumes oxygen, the air bubble starts to recede into the respirometer, and we can measure the rate of respiration based on the change in that reading on the 1 ml pipette. Okay, so for example, if this is all water right here and this is air, as cell respiration proceeds, you'd see that bubble proceed in this direction as the volume of gas decreases. So the first thing you would need to do is to set up an experiment where you have, we need to set up a control experiment. We need to set up an experiment where we're looking at organisms. And to do that, we need to do a displacement, water displacement, to calculate an equal amount of beads that we will use for our control. And we want to do the following. So you take 50 milliliters of water in a graduated cylinder, and you add 20 or 25 Ps to that. So just define what you're doing. So we're going to add 25 Ps, and then you measure the amount of displacement of water. So however many milliliters that rises for 25 Ps, you want to take another graduated cylinder and also fill it up to 50, and you want to add beads until it equals the same amount of milliliters displaced. So that'll give us an equal volume of P's and beads to add to our experiment, and I'll show you what I mean by that in a moment. So this is how we'll construct our respirometer. So essentially, again, it's a glass vial with a stopper and a pipette sticking out of it. On the back, we have these washers to weigh it down, because remember, you want it to be submerged in water. And then inside the respirometer, you'll see the following components. So you're going to see absorbent cotton balls all the way to the back that have a predefined amount of potassium hydroxide added to it. So for example, you can say 20 drops of potassium hydroxide to this absorbent cotton ball for students. And in front of that, and I'll explain what potassium hydroxide does in a minute, in front of that, we put a non-absorbent cotton ball that acts as a barrier between, the one that has potassium hydroxide and our organism that we're studying. In this case, this is the control, so there is nothing living here, but we have these glass beads, okay? And the glass beads control is basically going to control for pressure changes that would occur in the environment. So we're going to do this experiment side by side with our other experiment. In this case, we're studying germinating peas. So we're going to do it side by side with germinating peas. We've controlled for it by having the same amount of volume inside the respirometer. So essentially what we've done is we've controlled for pressure changes. We're going to control for this air bubble moving in response to pressure changes in the atmosphere. So I imagine that in different environments around the world, this would be a problem. For us here in Pennsylvania, it's not really a problem, but it does allow us to be more precise in our measurements of how the respiration rate, how fast the respiration rate is occurring. Okay, and then since this is occurring in water, we're going to have a thermometer in the water that shows us, we're going to keep this water at room temperature for one part of the study. Okay, so this experiment does a good job of asking students about controls and constants. Okay, so this glass vial, this glass bead vial that contains the beads is our control for pressure. Another thing I'd like to mention, that if you were actually doing this experiment, you'd set up a 5-minute equilibration period where the 2 vials, 4 vials, however many vials you have going, would essentially rest on a piece of tape that is the glass vial part where the organism and the control beads would be submerged in the water to let the organisms acclimate to that environment, that temperature, whereas the tips of the respirometer would not be in the water just yet. They'd be resting on the side, the edge, or on a piece of tape so that the experiment doesn't start yet. So you want them to adjust to that environment. And so there's a 5-minute adjustment period. So I want to go back and explain why we have potassium hydroxide on that absorbent cotton ball. So the experiment is essentially designed to measure the amount of oxygen that an organism like germinating peas would be consuming as it performs cellular respiration to meet its energy needs. The problem with that, as you recall from the Krebs cycle, is that they're also putting out another gas in response to that rate of cell respiration, that gas being CO2. So if we're creating equal pressures of gases coming in and gases going out, you're not going to see that bubble advance. So what we've done is we put potassium hydroxide on the cotton ball, and potassium hydroxide can take gaseous carbon dioxide and precipitate it into potassium carbonate. So really what we're doing by adding potassium hydroxide is we're dealing with CO2 as a gas and removing it from the equation so that oxygen can rush in, and so that the gas bubble, the air bubble, can proceed as a measurement for oxygen consumption. Okay, so now that we've removed CO2 pushing back on the air bubble, now you can just see the air bubble recede into the pipette as the organism undergoes cellular respiration. Okay, so this is a good lab to consider, the ideal gas laws that you learned back in chemistry. And I've never really seen the College Board actively ask about this question, which is not to say that they won't ever, but you should be able to understand the relationship between pressure, volume, gas number in this experiment. So we've actually controlled for this, and so we really want to see the relationship between temperature, pressure, and volume in this experiment. So the key part for us is if we keep temperature and pressure constant, the volume of the gas inside the respirometer is going to be directly proportional to the number of gas molecules. So as those, as oxygen is consumed in the respirometer, you'll get an idea of the number of oxygen molecules left over. So, essentially by the setup of the experiment, we've controlled for variables, and then you can see the relationship between those components of that equation. So again, going back to this, this design with the glass beads, controlling for pressure changes in the atmosphere and temperature, we've controlled for a lot in that experiment. Okay, so now we want to perform the experiment. So in a second glass vial, you'll put those P's that we counted, the 25 P's will go in. And this is where the non-absorbent cotton ball comes into play, because potassium hydroxide is caustic and can kill cells inside the P's. So we want to protect them by putting that non-absorbent cotton ball. So this is going to be our test subject. Okay, we're going to be testing germinating P's in various temperatures, right? So the variable that we're looking at is temperature for these organisms. Okay, so an important note here too, a lot of times teachers will run 6 vials side by side, 2 controls, 2 germinating two germinated P's, 1 at 25, 1 at 10, and then some non-germinating P's, some dormant P's at 25 and 10. I've removed that from this video. I'll just go ahead and tell you that, dormant non-germinating P's actually are performing cell respiration at a much slower rate. You probably would have hypothesized that anyway, but we're going to reduce your workload here and just ask you to analyze the experimental data for germinating P's at 25 and at 10. Okay, so how does this work? Well, let's run the experiment for the control. That is glass vials containing a glass vial containing glass beads. Okay, so there's nothing living in there. Cell respiration shouldn't be occurring. You shouldn't see advancement of that water into the pipette. So we shouldn't see that that air bubble recede unless there are pressure changes occurring. So we're just watching out in this experiment for pressure changes in the experiment. So I've put in here a table for you to fill out. So basically you're going to follow the air bubble, seeing if it changes at all at these time points. So when you drop the respirometer into the water, not when you drop, when you place it into the water, what is the reading on the pipette when you put it in there? So what is the initial reading? And then every 5 minutes for 20 minutes, you're going to take a reading of where that air bubble / water interface is. Now, I would hypothesize that in our lab, you're not going to see this bubble advance very much, maybe if a little tiny bit. So I'm going to ask you to take those readings. Here in this column, you'll basically subtract the change in that bubble's placement over time. So let's just go ahead and show you what I mean. Okay, so we're doing our control at 25 degrees Celsius, which is room temperature. I place the respirometer in the water, and the water rushes all the way into 0.8. So your initial reading at time 0 would be 0.8. And then I'm going to come back to this pipette tip every 5 minutes and read it. So when I come back 5 minutes later, there it is. I'll go ahead and tell you what this is. So I've actually done the readings for you for the control. This is 0.79. Okay, so it's moved a 100th space. Here it is, 10 minutes. It's moved again a little bit. We find ourselves here kind of at 0.78. 15 minutes, 20 minutes, and here's your table. Okay, so it hasn't really moved a lot. This, basically there's no respiration going on. We haven't really sensed any atmospheric pressure changes. So really, our experiment should be pretty much good to be read for the germinating P's. Now, what we'll do is we'll subtract 0.01 from whatever the reading is for P's at that point in time, just to control for that change. This change is essentially random change, just random moving. So to compensate for that, we'll subtract that at the end. I'll show you that in a moment. But let's go ahead and run our experiment for germinating P's in the respirometer setup at 25 Celsius. What is your hypothesis? What do you think is going to happen in this experiment? So go ahead and write that down. Here's your data table that I want you to fill out. So again, you're following the readings at every 5 minutes. You're calculating the difference between readings, and the corrected difference is you're subtracting the difference seen in the control glass bead respirometer for each of those time points. So you're getting more precise by subtracting that control data. Okay, so here's your data for germinating peas at 25. You place the respirometer in the water, it starts at 0.8. There it is at 5 minutes. There it is at 10 minutes, 15 minutes, and 20 minutes. So go ahead and fill out your data table here. I'm very specific in what I want you to do here. Okay, so you're going to take this data right here. Once you have filled out the readings, once you've calculated the difference between time points, I want you to subtract these numbers from those difference numbers, and that's your corrected difference. And by the way, that's the data that I want you to graph at the end here. Okay, so we're going to continue to run this experiment. We're now going to run our control with glass beads in our classic respirometer setup, but we're going to do it at 10 degrees Celsius. So we've lowered the temperature of the water bath that these respirometers are submerged in. So again, here's your data table, and here's your data for the control experiment. So there it is, 5 minutes, and I forgot to write that, but I'll go ahead and say it. So you place it in the water at 0, that's your initial reading. There it is at 5 minutes. There it is at 10 minutes. There it is at 15 minutes. There it is at 20 minutes. So there, I've calculated this for you, but I do want you to be able to read those. So here's your control data for the glass beads experiment, and I've calculated the differences there. Let's go ahead and look at germinating peas at 10 degrees Celsius. What would you hypothesize? Would that rate of cellular respiration increase or decrease? Go ahead and write that down. Here's the data table I want you to fill in. So here's germinated peas at 10 degrees Celsius at time point 0. So I put the respirometer in. The water bubble goes all the way to 0.75 at 5 minutes, at 10 minutes, at 15 minutes, at 20 minutes. So go ahead and calculate your data there. And I'm going to give you a head start on your graph as well. So looking at this graph, and by the way, here's an example of a title I'd like to see. Don't copy it. I guess I am giving you a good example of a title, but don't copy it. Come up with your own. So I'm calling this Reducing Temperatures Reduces Respiration Rates in Germinating Peas in the Respirometer Lab. So I kind of gave away the data, but you could see that coming through the experiment, right? Okay, so on the y-axis, we're going to plot our dependent variable, which is oxygen consumed in milliliters. So 0.2, 0.1, 0.2, 0.3, 0.4. Okay, and then on the x-axis, we're going to examine the experiment over time. Okay, so you should come up with two lines here. One line is going to talk about the data you saw for germinating peas at 25 degrees Celsius, room temperature. And the other line is going to show me what happens when you lower that temperature by putting ice, literally ice, into the water to lower it down to 10 degrees Celsius. Okay, so I've given you a head start on that graph. I know this is a longer video, but I hope this helped you to get a good head start in understanding respirometer labs and how they can show you cellular respiration rates in various organisms. We've been looking at germinating peas in this experiment, but really you can put anything that undergoes cellular respiration inside that vial. And I've seen some people do some fun and weird stuff where you put crickets and other sorts of organisms in there. So get an idea based on what you know about cellular respiration and metabolism rates in general. Get an idea of what you might see if you looked at different organisms of different masses, different sizes. Okay, and we'll talk about this in class.